Hello friends, today I am going to show you how we can do gene expression analysis in our environment using HR package. So the first step is to providing a high quality and well structured data set which contains number of genes, number of samples, and read count per individuals. Here I have two groups of individuals. The first group is related to normal, people with normal status, and the second Excel sheet file has a read count for individual with osteoarthritis or case and control. So I have two groups of individuals. As you can see, the number of columns or each column representing just one sample, and the rows, each row representing one gene. So I have genes on rows and samples on columns. So if I go through here, you can see I have 18 samples, normal samples, and I have about 23,000 genes on rows. So for example, this cell shows read count for gene A to M for first individual, and here shows for the second individual and number of count for third individual, and so on. I have the same scenario for my case or osteoarthritis samples. If you look here, you can see I have 20 samples and I have the same number of genes, and the order of the genes are completely similar between two samples, two data sets. So I need to read this data set in R and do gene expression analysis. So let's go in R and see how we can read this file and do gene expression analysis. Please don't forget to subscribe the channel, and let's go over the next step. Now we are in our environment and we are going to read our data as the first step. Here I try to annotate my script to be readable for you. As you can see, I am going to start by loading 5 packages. The first package is read.excel, which is related to reading the Excel file. If you have a .txt file, you can use read.table function in R to read your file. So the second library is HR, the main library I am going to use for differentially gene expression analysis, and the 3 other packages for visualization and data manipulation. So first I set up my past directory and try to read the data. So fine. As I showed you, I had 2 sheets in my Excel file. The first sheet was related to normal data, so I tried to read my normal data. You can see I was able to read it. And here the osteoarthritis data. So I was able to read both data. Then I am going to combine them to make a count matrix. So as I said, the first column was associated with the gene name, so I should ignore it. And for the both data sets, I had the similar column, the first column as the indicator of gene names. So I will ignore it and then use data.frame function to combine both of them. Okay, I did it. Then I am going to assign a name for each column. As you can see, my first data was related to normal, so I had 18 samples, and I had 20 case or OA samples, so I will use this function paste 0 to create names for the last 20 samples. So, and I assign the column names. Also, another thing I need to provide for this analysis, I should say the name of my genes. So, as I said, the first column was related to gene name or gene symbol. Here you can see I have a vector of gene names. And the third step is to create a DGE list for HR. This function accepts 3 components. The first component is read count. As I explained here, I created, and I have groups. So I say how many groups I have. Here I need to repeat normal 18 times and OA 20 times, and say I have 2 groups with normal indicator and OA indicators. Then the gene symbol or gene name. So let's run it. As you can see, I created a DG list as an input for HR analysis. So the next step is going to do filtering and normalization, which is very important to remove noisy read counts and genes, because we need to have informative data for this analysis. This HR has 2 functions, filtered by XPR, which after we perform this function keeps genes with sufficient expression across all samples. In other words, a gene is kept only if it has enough counts in a minimum number of samples within each group to be considered expressed. Also, this improves downstream differential expression testing by reducing noise and avoiding inflated multiple testing penalties. So let's do it. It was the first filtering. Then we need to do TMM normalization, which is another critical step we need to do. And let me press this button. Actually, this function compute a scaling factor that adjusts each sample so that expression distribution become comparable across the data set, across the case and control, which maybe some noise we have because of differences in sequencing depths between samples, compositional bias caused by a few highly expressed genes because they dominating libraries. So we need to perform this as well. So after these 2 steps, we are 1 step further to do the final step for gene expression analysis. Okay, let's go ahead. Okay, the next step is to do exact tests to actually quantify the D genes between cases and control. As you saw earlier, I did quality control, filtering and normalization. Even though filtering and normalization I done, it is considered best practice to ensure a fresh, clean DG list object again, because by doing again this kind of quality control, we can remove any remaining law expressed genes that may not meet statistical criteria, recalculate normalization factors to ensure no bias from removed genes, and also ensures consistency and reproducibility in the exact test workflow. So I would suggest you can do this kind of quality control again. And the next step is to estimate dispersion using exact test framework. Why we need to estimate dispersion? It helps to determine the precision of each gene's expression estimates. This is the first reason we need to do it. It directly affects the statistical significance in exact test. Also, it ensures appropriate modeling of biological and technical variation, because our package, HR package, is going to do a negative binomial model analysis, which requires estimating biological dispersion, which quantifies variability between replicates beyond a Poisson variance. So we need to estimate this dispersion per sample. And here, exact test function actually is a final step for this analysis, which compares normal versus case control or osteoarthritis in my study and quantify the DE gene for me. So let's push the button and see. Okay, as you can see, I got some results, and their HR has a very nice function, top tags. You can select how many top genes are you going to extract. Here I am going to have information for all of my DE genes, all genes, and I am going to do a p-value quality control or correction. We have, as you know, we have multiple methods for correction. Here I am going to use FDR, false discovery rate adjusting method for each gene. So let me push this and see the output. As you can see, I have a sorted gene list. Here I have gene names, log fold change, log CPM, p-value per each gene, and FDR. So I got the output. The final step right now, and we were able to get the output of the gene analysis exact test. And right now we are going to define two thresholds to pick the most important genes for our rest of analysis. So here I am going to define two thresholds. The first threshold is log fold change. I am going to select those genes with higher fold change than 1.5, and also those SNPs with 0.01 FDR value or corrected p-value. So by defining these two thresholds, let me hit the run button. I can classify genes in three categories: non-significant, upstream and downstream, or downregulated and upregulated and non-significant genes by these three lines of code. So, and also we can define this label for them and also add a column as a minus log FDR value for visualizing the results. Let's see the final output. Here you can see I have an extra column for down, up, and non-significant genes, and also minus log 10 FDR for creating a volcano plot. For volcano plots, I am going to define the colors for three different categories. I'm going to have a red orange for upregulated, deep blue for down-regulated, and non-significant genes also I would like to have a gray color. Or here I used the Ghibli palette. Is a well-known function for calling the beautiful colors for plotting. And also I prepared a piece of ggplot code for creating volcano plot. Let's run this part. Okay, here we have a very beautiful output. Each dot represents a gene, and also we have log fold change on x-axis, and we have a FDR on the scale of minus log 10 on y-axis. And also we can see we have three categories of genes: down-regulated, up-regulated, and non-significant SNP genes. Here you can see I have 272 upregulated genes and 283 downregulated, and about 13,000 non-significant genes, which is really great visualization of gene expression analysis. You can use this plot for your publications. And if you are going to write down your results, you can use write.csv and also write.table to write your result as a .csv file or .txt file. So I hope this video helped you to do some initial gene expression analysis. Just I should mention, sometimes we have more complicated models. For example, we have many covariates: age, sex, family, and different nested groups. So you can design your model. HR is able to help you to design different models. It's not too difficult. So, and don't forget to subscribe the channel.